Well, we're going to go ahead and get started. Again, thank you all so much for being here and joining us for this session of Empower Hour. This event's being brought to you by the Ohio University Alumni Association, where our goal is to advance the mission of Ohio University by engaging you, our supporters and alumni. So thanks again for being here. We really need you to stay involved with this great university and the university that you love so we can keep providing new and unique experiences to current and future Bobcats. I'm really excited to have you. And my name is Caleb Biesecker, and I'm the Associate Director of Alumni Experiences, and I'm going to be your host for the day. And we're just going to start by going over a few brief housekeeping items. And so to start, for your knowledge, I wanted you all to know that your camera and microphone is off, but that doesn't mean you're not able to communicate with us. We would love for you to be engaged and involved in the chat and participate there as much as possible. We're going to try and answer questions throughout and just address any topics in there and just keep a nice open dialogue going there. It makes things a lot more fun for everyone when you're engaged and involved there. Also, if you have any tech issues, feel free to drop those there, and myself or Barb Fiocchi on our events team is happy to help as well. Also wanted to make you aware we're going to be recording the presentation for today. So if you enjoyed it or maybe have to miss a little bit of it, go find it on the Ohio University Alumni Association webpage and share it with your friends and rewatch it if you want to as well. And then I'll provide another reminder. We do have a Bobcat code, or excuse me, a Bobcat store discount code in the chat for 20% off. So be sure to visit the Bobcat store and take advantage of that. And then lastly, I would just ask that you be on the lookout for our follow-up message after this event. It's just going to include a brief survey that you can take, and we just really value your feedback because we want to keep improving these events. And so we would really just ask you to participate in that. And then also there's a chance to see more programs that are upcoming, as well as a link to the new Ohio Giving page where you can support programs like Empower Hour, as well as student scholarships and other important things going on at Ohio University. So thanks for listening to that. Looking forward to this presentation. And now I'm going to turn things over to our presenter for the day, Dr. Doug Getz.
Thanks, Caleb. And thanks for everybody for joining us today. And thank you for the opportunity to present. My name is Doug Getz, and I'm a professor of chemical and biomolecular engineering here at Ohio University. And the title of my talk today is Development of Therapeutics that Target Malfunctioning Messengers. So I've had the good fortune of working with excellent faculty and students here at Ohio University over the last 20-some years. And I wanted to highlight them here before I got into the talk. In particular, Kelly McCall, who's in the Heritage College of Osteopathic Medicine, Steve Bergmeyer, who's the department chair for chemistry and biochemistry, and Jennifer Hines, who's also in chemistry and biochemistry in the College of Arts and Sciences. I've had many good students. They're all listed here. I'm not going to name them all. And I'd also like to thank the National Institutes of Health and Emergent Ventures for funding some of the research I'll be talking about today. So this first slide is a question I'd like you to read. And after you've read it, Barb will post the poll question 1, and I'd like you to answer that. So the question we're attempting to answer in my lab is related to COVID-19 as follows. Could inhibitors of the kinase GSK3 be efficacious in COVID-19 via inhibition of cytokine storms? So that's the question. And then Barb has a poll question for you. And I'll say up front that we expect a lot of these answers to be no. And I think that's what we're finding, that a lot of these are no. Some of those are yes, which is kind of interesting. That's good. Okay. So we got the final result there. And interestingly enough, it looks like 74% of the people wouldn't be able to discuss that, and 25% think they could. And I'll try to remember those numbers. And the goal of the talk is that when I flash this slide up at the end of the talk, that number goes from 74% saying they don't understand the question to hopefully 74% or higher even, although whatever it is, saying they can understand the question. So for the goal of the talk then is so for you guys in the audience to understand what the question is we're attempting to answer in my lab, the group of us. Okay. So the second poll question relates to cells. So the majority of the work that I do relates to at the molecular and cellular level. And I was wondering how many people have heard of cells? I'm guessing this will be more than 74%. And it looks like we're at 100%, which is good. So let me move this over. So 100% of the people have heard of cells. That's good to hear. So your body's made up of lots and lots of cells. In fact, you have over 30 trillion cells. And although all of you have heard of cells, I did want to highlight a couple of features of the cells that we'll be talking about today. So one way to think of a cell in your mind is if you take a little bit of oil and drop it into water, you'll notice, like olive oil into water when you boil pasta, you'll notice that the olive oil makes little droplets of oil within the water. And if you take that olive oil and take all the oil out of the middle of the droplet and make it water, so you just have a shell of oil on the outside, that begins to be the structure of a cell. So it's got an oil layer on the outside, which we refer to as the plasma membrane. And inside it is an aqueous solution, a water solution, that has a bunch of things we call organelles inside it. So you know we have organs, we have brain, we have a heart, we have lungs, but we have organelles inside the cell that do specific functions. I'm not going to go over all those today because we don't need to know about them, but the one I did want to highlight was the nucleus. And that's where all of our genetic material is stored. It's encased by a membrane itself, an oil layer, but inside it's the aqueous layer, and that's where all of our genetic material is. So that's kind of all the details we really need to know about a cell. It has an oil layer, which is the plasma membrane. It has organelles inside, and one of the organelles is the nucleus. Now we know we have multiple types of cells. We have skin cells, hair cells, muscle cells, and blood cells, which will be the focus of the talk today. Now we also know that all these cells aren't the same. You can see your hair cell doesn't look like like a blood cell, as you can imagine. And so one question is, how are all these cells different? Well, they're different because they have different distributions of the building blocks of the cells. So what are the building blocks of the cells? Well, roughly it's proteins, lipids, and carbohydrates. We don't really need to focus on carbohydrates. The lipids are what make up this outer layer. But the proteins are the major focus of the talk today. So we do need to know about those. And those of you who are vegetarians, if you eat tofu, that's how you get your protein maybe. And carnivores will eat meat, and there's a lot of protein in there. So you've probably heard sort of the layman idea of what a protein is. And the different distributions or different types of proteins that are turned on is what causes you to have different types of cells. Now, proteins play an important role, including structure, and what we'll focus on today, the mechanism of communication. So as I said, we'll focus a lot on proteins, and the central question is, how are proteins made? So again, in the nucleus, we have our DNA material. So the nucleus is shown here. We're expanding it from the cell, and it has a membrane around it. And it encases all of our chromosomes. And you may remember those as being X and Y. And so I've listed a bunch of X chromosomes here. And these are coils of DNA. And that DNA contains the instructions for proteins. So how does that happen? So I have my DNA in the nucleus. The DNA gets transcribed into mRNA, and the mRNA gets translated into protein. So it's a two-step process to go from DNA to protein. I go DNA, RNA, RNA, protein. And the way I get different kinds of cells, and more importantly for this talk, how cells respond to their environment, is by changing their expression of proteins. And they do that by activating certain regions of the chromosomes. So they're not expressing all the DNA in your body all the time. You're only expressing certain sequences within your chromosome, certain parts of the DNA that leads to different cell types and the response of your cells to the environment. Now, as I said, you have lots of different types of cells in your body. And today we're going to be focusing on blood cells. And we can group these into red blood cells and white blood cells. And here's an image here from the National Cancer Institute. Here's your red blood cells. They're sort of disc-shaped. And here's your white blood cells that are spherical. There's other cell types here, but we're not going to talk about those. So your red blood cells are really important because they carry all the oxygen around, right? And you probably know that they provide the oxygen to the tissues. Although they're very important, that's not going to be the focus today. We're going to focus on the white blood cells. And they're important for wound healing, fighting infection, and response to invading organisms. And there are multiple types of white blood cells. We can divide them up even further into lymphocytes, which you may have heard of because of HIV/AIDS, and another group called the macrophages, which will be the focus of our discussion today. So the macrophages play an important role in this process called the innate immune response to invading pathogens. And this is actually a really cool process. So as we sit here today and listen to this webinar, we're under attack by pathogens. You've got bacteria, you've got viruses that are trying to attack your body and set up shop and reproduce. But fortunately, you have your innate immune response, which is the first response is trying to fight these invading pathogens. And the key to that are the macrophages. And this is in contrast to your acquired immune system or your acquired immune response, which activates because you've already seen a pathogen. So for example, when you get a vaccine, that's what happens. You get the vaccine and you build up an immunity to that pathogen represented by the vaccine. And that becomes part of your acquired immune response. But your innate immune response is sort of the thing that just right when the bacteria arrives, it responds right away. You don't have to have a clonal expansion of these cells to get the response. So that's why it's called like the innate immune response. Now, how does this happen? Well, the macrophages, a subset of your white blood cells, detect these pathogens, bacteria and viruses, because the pathogens shed something called pathogen-associated molecular patterns, or PAMPs. And we'll only use that term a couple times. So the things that are invading your body, they're shedding things, and then the macrophages recognize that, and then they generate a bunch of proteins. And this initiates a vigorous response to eliminate the pathogen. So with this part of the introduction of the talk, we can put a lot of these pieces together. And that is, if we have bacteria here, here's an image of bacteria, this one's sort of rod shaped. It invades the body, it releases PAMPs. These PAMPs kind of look something, let's say like this, some sort of molecular signature molecule, a molecular signature. And that's recognized by the macrophages that then generate a bunch of protein and initiate the innate immune response. So that's all pretty clever, right? That's how your body starts fighting a bacteria, or I could substitute this for a virus, and that's kind of how all this happens. But if you step back and think about it for a little bit, you realize that a major piece is missing. And that major piece is right here. So this signature molecule from the pathogen is typically outside of the cell, so swimming around out here. Yet I need to make proteins, which if you remember from the first few slides, happens because I transcribe DNA to RNA and then RNA to protein, and that all happens in the nucleus. So that's not only inside the cell, but it's inside the nucleus, so it's across two of these membrane layers, the plasma membrane and the membrane to get into the nucleus. So how in the world does a macrophage take this signature molecule and turn it in to protein generation? Now, to understand that, it's helpful to consider the Pony Express. So the next question is, how many of you have heard of the Pony Express? And if all of you are from the States, my guess is that we're going to get 100% on that one. Oh, wow, we have some that have not heard of the Pony Express. That's interesting. Okay, good. So I can teach you a little bit about that. So the Pony Express was a way to send messages using a sequence of messengers, across the Western United States. So this was in about 1860, and it lasted for about 18 months. So one of the terminuses of the Pony Express was Sacramento, and another one was a place in Missouri. And the idea was, how are we going to get mail and information between these two sites that spans half of the United States? Well, one idea would be to send a pony with a rider. Remember, there's no internet, there's no telephones, no cell phones, things like that, and have them try to ride this entire distance. And then once they got there, collapse and hand them the message. But a better way was to say, well, let's have one rider ride about 50 to 75 miles, hand it to the next rider, then this person can, spend the night and then head back. Then the next messenger rides for 75 miles, hands it to the next one, rides for 75 miles, hands it to the next one, and on and on and on. And that way we can get the message across the Western United States. Now, one thing that this could be really useful for is it could be used for an emergency response communication system. So for example, if we're in the West and we had an earthquake, we could say, well, if we have an earthquake, we really need other people to know because we need some help. We need help from other people to send supplies to respond to our emergency. So if we have an earthquake in California, we'll send the message through the Pony Express. That tells people in Missouri, initiate some building and ship some supplies out to us because we have this emergency in California. So help is on the way once the Pony Express transmits this message. Now, it turns out your cells have a system that's analogous to the Pony Express. It's called cell signaling. And it's an elegant and intricate mechanism for cell communication. And I've depicted this generically here. Well, it's actually specific for one particular signaling process. I took this from the cell signaling technology site. And each little node in here is sort of the transfer from one Pony Express rider to the next. So overall, we have the cell. This is just a portion of the cell. We have our plasma membrane. We have the nucleus here. Again, there's a membrane that surrounds the nucleus. We have the DNA. And we have all these messages going within the cell as depicted by these arrows. All right, so a network of Pony Expresses. So now, can we use this as an emergency response communication system? And the answer is yes, that's what your cell does. So in particular, your macrophage, which again is one type of white blood cell, it uses it to respond to pathogens. So we've got our plasma membrane, we have our nucleus, we have our DNA here. And then the question was, if it sees parts of bacteria on the exterior of the cell, how does it generate proteins in response? Well, these bacterial signature molecules are detected by the macrophage. That in and of itself is kind of a miraculous process, but it gets detected. That tells this molecule to change a little bit. That tells this, then it in turn tells this molecule to change. Then it tells this molecule to change a little bit, this molecule to change a little bit, this molecule to change, this one to change, this one to change. Finally, this one goes and crosses the membrane, binds to the DNA, and that leads to mRNA being generated. The mRNA is generated and makes protein. The innate immune response happens, and help is on the way. So this is the mechanism by which the macrophage responds to a pathogen, very much like the Pony Express. Now, what are the messengers in our cell? The messengers are what we call kinases. They're the pony and the rider. And they deliver the message from outside the cell to inside the cell. They're the messengers. So just to be clear, I've depicted, or this image has depicted organelles here, but these things here are not organelles. They're proteins, a lot of which are kinases. Now, some of them aren't, but for today's talk, we're just going to focus on the ones that aren't kinases and make it simple and say the communication happens because these pony express riders, kinases, transfer the message through the cell. And we have to have one more scientific detail, and this will be the last one. It's sort of an introduction to the chemistry or biology of it. So how does it transfer the message? Well, some of you may have heard of ATP. It's the energy molecule of your cell. It has 3 phosphate groups. I've depicted here as Ps. And what the protein kinase does is it takes a phosphate group off of the ATP and attaches it to another protein. So it's a simple reaction. You can just think of it as 3 Ps here, phosphate groups. Don't have to worry about whether they're phosphates or whatever. They're just Ps. And then it's an enzyme, and it takes the P off here, puts it on the protein, and now you have ADP. Triphosphate here, diphosphate here. So it's a very simple reaction. And that's what's happening in all these arrows. That's kind of the handoff of the message between the pony express riders. It's phosphorylating this protein that then may phosphorylate the next protein and phosphorylates the next one, on and on and on, transferring the message. So then we transcribed our DNA. Now, we have to go back to our pony express analogy one more time.
Hey, Dr.
And
had a quick question for you that popped up. Wanted to ask you. Someone asked, how long does the process take to go from DNA to RNA and kind of to that protein then?
Oh, I got a bunch of questions here. Sorry, I didn't see them. I think it's pretty quick. That's typically in our experiments, we can stimulate with something and see protein in a few hours. But so I would say as short as an hour or two to go from, stimulating to get the DNA to make the RNA, and RNA to make the protein. You can see that maybe an hour. Typically, you can see RNA in probably 30 minutes, but it can take longer as well.
Great, thanks.
Yeah. Okay, so the Pony Express, okay, going back to this. So we have the Pony Express and it's all working well, but then what happens if a rider goes rogue? So we have a rider in Wyoming that decides that they're kind of bored, and the rider thinks it's fun to send signals about earthquakes in California. So there's no earthquake in California, but the rider's sitting here in Wyoming kind of bored. And so they get up one day and say, I'm going to send a message that there's an earthquake in California. So the rider does that, and this message gets transferred all the way back to Missouri. And the good people in Missouri say, oh my gosh, they're in desperate need in California. Let's start building some stuff. Let's all head out there. Let's help them. And the rider's having so much fun with this that the next day, the rider does it again. Waits a couple more days and does it again. And so eventually these people in Missouri are like, oh my gosh, it's just crazy in California. Let's start taking parts of our house down and ship them out to them. We can get by without this barn. And eventually we have a pathology of activity that's really not going to be good for Missouri. And once California gets all this stuff, it's not going to be good for California either. Similarly, we can have rogue cell signaling leading to disease. So if I have a signal from outside the cell that's interpreted as a pathogen, or more likely, if one of my signaling molecules, one of my kinases, goes a little bit rogue and gets phosphorylated and starts phosphorylating everything else, then I can get DNA made to RNA, RNA made to protein, and that can lead to diseases, to pathologies, cancer, arthritis, Alzheimer's disease. And this is pretty true. You name a disease, and give me half a day or so, I can finally probably find a paper that relates it to some sort of pathological kinase signaling. In particular, COVID-19 may involve pathological cell signaling. So going back to the kinases then, given that aberrant cell signaling can cause a lot of disease processes, we know that kinases are really, really, really important. And that's emphasized in these next two slides. There's over 500 protein kinases that have been identified in the human genome. Now we have to be a little exacting in our wording here. So by this, I don't mean every cell has 500 copies of the same protein kinase. We have 500 different types of kinases. So 500 different pony and riders that are each different. And each one of these does a different task. It phosphorylates a different set of proteins, behaves a little bit differently. So they're all unique. 30% of all the proteins you have, and if you think about it, you're made up of a lot of proteins, are modified by kinases. So they're ubiquitous, they're acting everywhere. And as we said in the last slide, there's abnormal function leads to a myriad of diseases. Abnormal signaling is often traced back to abnormal protein kinases. Not surprisingly, given all of this, there's a huge effort in the pharmaceutical industry to find kinases, the molecules that can inhibit kinases. And I'm just going to step through this slide, just read it because it's said probably pretty succinctly. But the take-home message is there's an expanding and highly successful global effort to develop kinase inhibitors into therapeutics. 30% of current R&D spend by pharma is focused on the development of kinase inhibitors. 75 drugs targeting protein kinases have been approved by the FDA for clinical use. The global market for kinase therapies is about 20 billion per year. And there's over 100 small molecule kinase inhibitors that are in late stage of clinical development. Given all this activity, there's only 10% of the kinases that have been studied in detail. So that's quite surprising. But remember, there's 500 kinases, and remember, there's 500 kinases, and only 10% of them have been studied. So only 50 of them have been studied in detail, leading to all of this activity. So it's definitely anticipated that over the next decades, there's going to be a lot of effort by the pharma industry to identify kinase inhibitors for all the other kinases that we have yet to explore. Now, when we think of developing inhibitors for kinases, we recognize right away that there's a significant challenge. Going back to our schematic of how a kinase works, remember, it takes the phosphate group off of ATP, puts it on a protein. So all kinases, or the majority of them, bind to ATP, and they take the phosphate group off and put it on a protein. So not surprisingly, even though they are all distinct, they're all very similar. So a major challenge in finding kinase inhibitors is to find ones that inhibit, ideally, only one kinase. And this is challenging because all the kinases behave similarly, and not surprisingly, are all structurally quite similar. So this sets the stage for the first paper that we did that I'd like to talk about today, just a couple slides from it. And the title of the paper was Identification of a novel selective and potent inhibitor of glycogen synthase kinase 3. Okay, and this appeared in last year in American Journal of Physiology, 2019. And there were a variety of people that contributed to this work, but I wanted to highlight Nora, who was a grad student in my lab at the time, who did a lot of the heavy lifting for this project. In fact, there's a good chance that this project wouldn't have been completed without her effort. She's now a scientist at Elanco in Iowa. So GSK3, you don't have to remember all these words here, just remember GSK3, was identified 35 years ago. So this is one particular type of kinase. Remember, we had 500 of them. This is just one of them we're focusing on now. When it was identified 35 years ago, it was shown to play a role in glycogen synthesis, or glycogen synthase was what it's named after, but it's glucose metabolism is essentially the role it was playing. Since its identification, it's been shown to play a critical role in various cellular events, and it's been shown to phosphorylate over 40 substrates. Not surprisingly then, it's been implicated in a whole host of diseases. So that makes GSK3 a very attractive target, therapeutic target. Now, we have an ongoing compound discovery program. Ongoing, I mean, it's been going on for like 10 years or so. And through this process, we identified a compound which we refer to as COB-187. And we found through this process that it appears to be an inhibitor of GSK3. So this is a compound, if you want to talk to your friends about it, it's a small organic compound. That's the class of drug it would be identified as, not a drug yet, but the therapeutic we're working towards is a small organic compound. And when we discovered that it was an inhibitor of GSK3, we actually didn't even know very much about GSK3. Some of my colleagues did, but we weren't actually seeking to find an inhibitor of GSK3. It just turned out through our screening process, we found out that it inhibited GSK3. So as I said, GSK3 has been around for 35 years, and not surprisingly, there are a lot of inhibitors of GSK3. One of them is called tiglusib. Another one is one you may have heard of called lithium. Lithium is used for bipolar disease, some of you may know, and it turns out it's a very weak inhibitor of GSK3, and that is in the clinic. But all the specific inhibitors for GSK3, I mean, it's on the market, it's actually used in the clinic. But all the specific inhibitors for GSK3, like tiglusib, they're in clinical trials, but they're not actually approved for use yet. But tiglusib is one of the most clinically advanced compounds. So here's our compound, here's tiglusib. So we sort of happened upon the fact that it's a GSK3 inhibitor. And so then, because we're kind of excited about that, and we thought it might have some use, we applied to the National Institutes of Health to support our research. Well, they came back and said, yeah, that looks kind of interesting, but you never compared it to tiglusib. And we thought, oh, that's a pretty good point. So we learned about tiglusib, we brought that into our lab, and then we decided to compare them side by side. And we're a little pessimistic about what the results would be because tiglusib was already in clinical trials and we just had discovered our compound. And so we compared them in what we call a kinase screen. So I have to take a couple minutes to explain this. Again, here's our reaction. Kinase plus substrate plus ATP gives ATP, ADP, and substrate. And companies have developed assays for this, where you can easily detect if your compound inhibits this reaction. At the time we did this screen, there were 404 unique kinases available in a kinase screen. Remember, there's 500 kinases we think there's 500 kinases in the human genome, over 500. But at the time, they only had 404 that were available. And I see somebody raise their hand. And maybe I can figure out how to do that. Caleb?
Yeah, you can keep going, Dr. Getz. If they have a question, they'll type it in. I think it was by accident.
Oh, okay.
Thanks.
Thank you. Sorry about that. Okay, so we had 404 unique kinases of the 500 we could screen, but still that's a lot, right? That was pretty good at the time. And so you give them the compound and they screen it against all these kinases, and they're in these individual blocks, okay? And the results come back like this, kind of reflecting the blocks that they did the experiment in. Each one of these, I hate to use the word cell because we're using it 2 different ways, but cell as in an Excel spreadsheet. Each one of these cells represents a different kinase, okay? And the color in the cell represents how much inhibition your compound imparted. So if it's blue, you really don't have much inhibition. If it's red, you've got a ton of inhibition. If it's white, you have a modest level of inhibition. So with our compound, you can see that it hit 2 of these kinases. Turns out this is GSK3 alpha and beta at a very high level. So there's a lot of inhibition of GSK3 alpha and beta. There's 2 different versions of GSK3, alpha and beta. And the rest of these aren't really affected except for these 2 here. Now with tiglusib, surprisingly enough, this isn't surprising. It hits GSK3 alpha and beta. It's touted as a GSK3 inhibitor. Interestingly enough, not as much on this isoform of GSK3 versus the other one. But yeah, we do see inhibition of GSK3. But over here, we also see it's inhibiting a lot of other kinases. And so it doesn't appear to be as specific as COB-187. And there's a reason why this could be, but I'm not going to go into it today. Now, the next question you could ask is, how potent is it relative to tiglusib? And that's the results of that assay are shown here. So we have GSK3 alpha and beta. As I said, there's 2 different versions of it. You don't have to worry about that too much. We'll just focus on one of these. This axis gives your percent inhibition. This axis gives you the concentration of inhibitor. And the red line is for GSK3, or I'm sorry, for COB-187. And the yellow line is for tiglusib. The way to compare the potencies is simply to draw a horizontal line from, let's say, 50%. So this is where I get 50% inhibition, and find out where it intersects your curve. So if I come over here, I get, oh, okay, if I have this concentration of COB-187, I can get 50% inhibition, and that concentration is just by going down and dropping a perpendicular down. So it's in the 10s or so nanomolar range of the compound will inhibit 50% of GSK3. In contrast, tiglusib needs to be up in the thousands of nanomolar range to inhibit the same level of GSK3 that we get with our compound. So in summary then, from this paper, there's lots of other information in the paper, but in summary, COB-187 is a selective and potent inhibitor of GSK3. So all those studies that were exciting to us anyway, were done at the molecular level. We just had the kinase, all the various kinases, and our compound, and we did it inhibit this molecular activity. Well, obviously, you want to find out how it works in cells. So that's the question we turn to next. Does this compound work in cells? Okay, and for this, the variety of different diseases we could have looked at. Remember that one slide where I said GSK3 is involved in a myriad of diseases? It's been implicated in it. But what we decided to focus on was the cytokine storm. So to understand what that is, we have to return to our signaling cascade diagram and look at it in a little bit different light. So remember that this signal transduction pathway that we're showing here is certainly a positive. It's the reason you and I are alive today. If we didn't have it, we'd be overrun by bacteria or viruses and be dead by the end of the day, probably, or at least at the end of the week. Okay, so it's a good thing. We're responding to components of bacteria. We're generating cytokines, and we're inhibiting these if they start to get out of hand. Now, the other piece of this is that these proteins we generate are called cytokines. All right. The ones, I mean, there's a lot of different proteins, but I want to focus on ones called cytokines. So I'm just changing this a little bit from the generic name proteins to cytokines. And one thing I left out before is these cytokines actually are very potent. They can come up and bind to the cell and cause a secondary signal transduction mechanism. They can bind to other cells and cause signaling in other cell types. Or you could have tertiary signaling where you get another set of cytokines that come out, and on and on and on. So you've got this cascading event, and that's a good thing usually when it's fighting infections, if it's started to, the bacteria is taking up shop in your body. But what can happen is sometimes those pathways don't shut off, or they overreact, they hyperreact to the pathogen. In that case, we get something that's termed a cytokine storm. So in summary, the cytokines are needed for fighting infection, but cytokine storms can occur where we have generation of unneeded and unwanted cytokines. This can happen in response to pathogen invasion, bacteria or viruses, can cause damage to tissue, organ failure, and death. And unfortunately, a lot of us are probably familiar with that phrase because of COVID-19. Tissue damage, organ failure, and death. Now, it turns out GSK3 has been implicated in bacterial-related cytokine storms. So having observed all that, we asked, could COB-187 attenuate bacterial-induced cytokine storms? That's our hypothesis. And this was a paper we submitted at the beginning of this year, and it got accepted and published in the summer. Modulation of LPS-induced inflammatory cytokine production by a novel GSK3 inhibitor, essentially our compound asking if it will inhibit cytokine production by components of bacteria. And again, Nora was critical to this paper. She's the first author, and I greatly appreciate all of her effort. So I only have a couple of experiments that I'm going to describe, or actually one experiment and 2 assays from it. But because it gets the point across without wading through all the data that's in the paper. So we want to find out if our compound attenuates LPS-induced cytokine storm. LPS is the molecule from bacteria we're using to mimic the bacteria or to mimic the signal that generates the cytokine storm. So LPS, just think of it as the component from bacteria. So how do we test this? Well, right here we have what we call a multi-well plate. And many of you have been watching the news, I would think, the excitement with the new possible vaccines for COVID-19. And if you look closely at those clips from the news, you'll often see something that looks like this being used in the laboratory. They're called multi-well plates, and oftentimes they're in robotic devices, and they've been kind of moving in and out of the robotic device, and fluids are being dispensed into them. Well, these contain a biological sample. And so what it allows you to do is you have little replicates of your biological sample, in our case, macrophages, in each one of these wells, and allows us to treat the various biological samples with different items, in our case, LPS, plus or minus the compound. And so everything's right next to each other. We can compare what happens without LPS to what happens with LPS, what happens with LPS plus our compound, and things like that. So it's called a multi-well plate, and that's what we did. We have this multi-well plate. We have samples of macrophages in each one of the wells. We had LPS in the presence or absence of our compound. We wait a while. Somebody was asking about that. We waited 4 hours, I believe, for this one. Then we harvest the cells. And then we look at the protein expression that we have, as well as the mRNA expression. Because remember, DNA goes to RNA, RNA goes to cytokine. The particular protein we're looking for is cytokine. So if we've turned anything on by adding LPS, if we induce cytokines, we should see an increase of RNA for cytokines and an increase for the proteins for cytokines. So here's the result at the protein level. There's a lot of details here. Don't panic, I'm not going to go through it all. We can focus just right here on what we call the blots. And what these blots represent are the expression of various cytokines that are listed here. So if you see 2 black dots, these black dots here are positive controls. They're not the cytokine. That just tells you the blots work. They're in replicate. That means the protein is present in your solution that you added. So we take the solution off the cells and add it and ask, is the protein present? And if we see a black dot, the protein's present. If it's blank, we don't see the protein. So let's focus on the dots around number 1, which represents CCL3, a particular cytokine or chemokine. Let's not do that. So if we have no treatment, we don't see this cytokine expressed, which is what you might expect because the cell shouldn't respond because there's no bacteria. However, if we take a component from a bacteria and add it, we see two dots representing CCL3, telling us that that protein is indeed expressed if the cells are exposed to LPS. Whoops, sorry. Cells are exposed to LPS. Sorry, that was a critical one to have the pointer on. Sorry about that. This is item 1 here. No treatment. We don't see anything. It's blank. LPS, we see the protein expressed. And now what's exciting is we add more and more of our compound. We see these black dots diminishing all the way up to our highest level. We see no expression of the protein. It's very much similar to the no treatment control. Now, as a gestalt, you can kind of quickly look at that and say, huh, I see a similar pattern with other cytokines listed there. And that would be correct. So we get a bit of a cytokine storm when we add LPS, and our compound reduces the expression of the cytokines. Now, what about the mRNA level? If we saw protein, we certainly should see the mRNA because that's a prerequisite to the protein expression. Now here we've listed it in a table. Here's our gene symbol for the various cytokines. There's a bunch of different names of the cytokines. They're all different molecules. They're all inflammatory molecules, or part of the inflammatory response or the immune response. And if we treat with LPS relative to background, we see 1 order of magnitude, 2 order of magnitude, 3 orders of magnitude, or even 4 orders of magnitude increase in the expression of these cytokines. Okay, so we definitely see a cytokine storm induced by LPS, which is what we should see. We expect that. But if we do it in the presence of our compound, you see a dramatic decrease in the expression of these cytokines. So what that means is COB-187 is able to, that abates the LPS-induced cytokine storms fairly dramatically. And that was really the take-home message from that paper. Okay, now this all brings you up to date of what was going on in the lab with the group of us all the way up to January of this year. And this leads into COVID-19. So as we know, it's the pandemic ravaging the world. The full name would be coronavirus disease 2019. It's caused by a virus called SARS-CoV-2. And it most often affects the respiratory tract, the nose, throat, and lungs. And many people have limited symptoms, but unfortunately, people can have very severe symptoms. Some mostly recover. There's no set of people, they mostly recover, and then they take a turn for the worse. In fact, there was a New York Times article in the early part of the pandemic, I can't remember which month it was, like January or February, that talked about COVID-19 and cytokine storms. In particular, what they profiled were 2 women in China that had COVID-19, and they both recovered and both went home. One of them never came back to the hospital, but another one came back to the hospital and had a horrible experience. And they described it as her suffering from cytokine storms. And since that point, there's been a lot of different information, papers out there implicating cytokine storms in severe cases of COVID-19. In fact, some people are actually suggesting using compounds that abate cytokine expression as a means to treat the cytokine storm that can accompany severe cases of COVID-19. So what this is now, now we're standing here and seeing all these articles, but I read that article in the New York Times in early January, and I kind of thought, but wait, we just published a paper about our compound being efficacious in cytokine storms induced by in a bacterial model. And now we have cytokine storms due to a viral model. And there's overlap between these pathways. And that's shown here. Again, returning to our signal transduction cascade, we were highlighting before the bacterial component of it. And this is what happens here. But if you trace all these lines, and first you draw a circle around the ones related to the viral response, those are all here. and then trace lines between the bacterial and the viral, you'll notice a subset of arrows that are common to both viral and bacterial cytokine expression, or viral and bacterial induced cytokine expression. So that means if you have a compound that inhibits cytokine expression due to bacteria, it could actually be efficacious in viral induced cytokine storms. So that was our hypothesis. So after I read that article in the New York Times, I got together with the team and we said, maybe these GSK3 inhibitors could work in COVID-19. And there's a bunch of GSK3 inhibitors out there. Like I said, it's been around. GSK3 has been known for about 35 years. So not surprisingly, there's a lot of inhibitors that have been discovered already. And some of them are in phase 2, phase 1 clinical trials. So perhaps if we could find evidence that these are efficacious in viral-induced cytokine storms, perhaps they could be efficacious in COVID-19. And that was the hypothesis we presented to Emergent Ventures, and we received funding for that, and we explored that over the last 6 months. So that leads me finally to the last question, the last slide, where I've listed the same question I had before. Could inhibitors of the kinase GSK3 be efficacious in COVID-19 via inhibition of cytokine storms? And the question is, would you now feel like you could discuss this at some level, educated level, with your colleagues or with your friends? And so we're getting the majority, they're saying yes, one that's saying no. Sorry about that to the one who's saying no. Okay, that's looking pretty good. Well, thank you for answering that way, even if that wasn't the truth. That's good. And that is the final slide. And thanks for your attention and making it through this long talk. And I'd be happy to answer any questions that you have.
Yeah, thank you, Dr. Getz. And I'll let a moment go by for some questions to go in there. But there was one question that someone asked, and it was kind of specifically around the COVID-19 and research being done there. And so they were curious, with an available vaccine coming soon, how quickly kind of your research could make it to the clinic and have an impact, or if you see that having any effect at all.
Well, that's a good question. So right now, they're using dexamethasone. You probably know that, most people now, because the president got it at one point. Dexamethasone is I think they're using it for this kind of the same reason. They're trying to tamp down excessive inflammatory response. So if they get a vaccine, that is a game changer. You're going to want to have the vaccine, and you would not need this as much. Yet still, some people may get it, and some people may overreact, and you still might want to do it. In general, also, this sort of idea of cytokine storms happens in, infections by different pathogens, like I said, bacteria, different viruses. So different bacteria, different viruses. And you may have heard of this process called sepsis. It's actually one of the major killers of people that go to the hospital. These cytokine storms are intertwined with this whole process called sepsis. So even if you get a vaccine for COVID-19 and we get it all in March and the whole thing goes away, which would be absolutely great, there's still a reason to question, how can you tamp down on cytokine storms? In fact, the idea, the complication of sepsis has been around for a long, people have known that for a long time, obviously, or understood it for a long time, but there's still really no good treatments for sepsis. It's actually a very vexing problem. Turns out it's also a problem in horses. It's one of the main reasons young, newly born horses end up in the veterinary clinic, is because of sepsis.
I'm not seeing many other questions come in, but I'll just ask one more of, I guess, kind of what next steps are for you and your team in this research process?
That's a good question. So I didn't present any of the data from our COVID-19 research right now because it's all very preliminary. So one thing we're trying to do is nail down our preliminary findings. We are definitely getting hints that our hypothesis is correct. So on the COVID-19 front, really it's just solidifying the data we have now, trying to do all the controls we need to do to make sure what we're seeing is due to what we think it's due to. On the more generic side of what are we doing with COB-187, our next paper is going to be about the molecular mechanism. How does it actually do this on a very detailed level? It's a very interesting study, but also a very hard one. And then we have a grant application out to try to use the compound, as I said, for horses. So that's another thing we're doing. And that would actually, it's not to actually treat the horses yet, it's to set the stage to do tests in horses. So we'll get material from horses, see if it works, figure out ways to detect the compound in horse serum, and that'll lay the groundwork for eventually testing in horses.
Not sure if you have your chat pulled up, but you might
Yeah,
be able to
I got
see
it.
questions. Yeah.
Okay, so a couple here. What is your gut about receiving a vaccine within the next year as far as safety? I'm not really an expert in vaccines, but I can give you my sort of pretty much armchair quarterback view of that. I would say there's part of me that says, this is, a lot of you could just answer this the same way. We get a new vaccine for the flu every year, right? So in some ways you could say, should it really take 8 years? Now you look at things like HIV, AIDS, and Zika and all that, you're like, well, that took forever, right? So I would, if the vaccine comes available, I will take it. So I think they're going to be good. Francis Collins was on the news hour the other night. Of course, one person presenting it doesn't mean it's going to be good, but he sounded trustworthy. I think I'm pretty positive about it. I feel like I think it's going to work out. I sure hope so. I think most of us are getting tired of being on COVID-19 lockdown. Kinase inhibitors used in autoimmune disorders. I should know the answer to that one. If I were to get to guess, I would say yes. Let me think about that. Autoimmune disease that I know the most about is colitis, and they use an antibody for TNF alpha. So they're targeting the cytokine directly. They're not targeting the generation of the cytokine, which would be more of a kinase approach. I actually, unfortunately, don't know the answer to that. In some sense, yes, I think they could, because my guess is, well, we were doing a study for a while on Graves' disease, which is kind of an autoimmune disease. And we were finding hints that GSK3 should work. So actually I have data in my lab that says it would work for thyroid-related autoimmune disease. So yeah, I think so. And my guess is if you did a literature search, you'd find kinase inhibitors, at least in clinical trials for autoimmune disease. Good question. Can mRNA vaccine prevent cytokine storm? That's an interesting one. So I think the person may be referring to the fact that Moderna, I think that's how you pronounce it. Interestingly enough, if you look at that name, it ends in RNA. The reason they've chosen that is it's emphasizing the fact that they're using RNA as the vaccine as opposed to a proteinaceous vaccine, which is kind of what was typically done in the past. That would be the first example of somebody using an mRNA vaccine. So they're obviously not targeting the cytokine storm. So this question is, could I develop an RNA vaccine to not cause a acquired immune response, getting ready for that, but for a cytokine storm? Hey, that's an interesting idea. I don't know. I have to think about it. It's a clever idea.
Well, I think we might make that our last question, though. And if you have any ideas that come to mind, I'm sure um, you can reach out, or they can reach out to you and try and get more details on that. It is an interesting concept. When they put that question there, I was like, oh, that's interesting. But we are out of
Yeah,
time.
they're saying no, probably. That might be right.
Yeah. But I am going to
Thanks.
cut us off there.
Okay.
Dr. Getz, yeah, thank you so much for your time today and the presentation. It was really interesting. I think it was awesome to hear about the students that have impacted your research, but also just the potential impact this could have on society, whether it's related with this pandemic or not. It just seems really, really neat and could have a positive impact on society. And it's cool to see OU kind of leading the way in that. And so we really appreciate your time and how you help Bobcats. And then I just want to say thank you to everyone that attended today. And just thanks for joining us for Empower Hour. And as a reminder, it goes on bi-weekly. We've got 2 more going on this semester, so be sure to join us. Going to be some awesome presenters. Don't forget to go to the Bobcat store, use your 20% off code that was at the beginning of the chat. You're welcome to look back and go find it there. And then be sure to fill out the survey that follows up. So thanks again. Hope to see you all at a virtual event again soon. And thank you again, Dr. Getz. Really appreciate your time.